In just 100 years, the world's population has grown from 1.5 to 6.1 billion. A result of this increase in population is that an increased stress has been placed on agricultural land, energy, and water in order to provide an adequate food supply. According to the World Bank and the United Nations, around 1 to 2 billion people are currently living under the poverty line and are malnourished. Crop and livestock disease are some of the greatest contributing factors leading to crop failures each and every year. This damage significantly diminishes crop yields and lowers farmers' incomes. Researchers at the University of British Columbia, led by Oliver Dong, a current doctoral candidate working at the Michael Smith Laboratories, have discovered an important class of genes in plants. These genes are responsible for plant protection against pathogens that can cause crop diseases. Pathogens are foreign organisms to the host species, such as a virus, bacteria, or insect that can cause disease. Scientists extracted gene sequences from a plant known as Arabidopsis and tested for changes in a class of 3 related immune system receptors known as TNLs. Researchers wanted to see how changes in these receptors would affect the immune defenses of the plants. Receptors are structures in a plant that can recognize and bind to a pathogen such as a bacteria after it has invaded the plant. Once the receptor binds to the pathogen, it can signal an immune response in the plant. The plant's immune system will try to kill the pathogen after it has been triggered by the receptors. But some of the pathogens may be able to resist the immune response and go on to cause disease. Researchers discovered that by inactivating one of the 3 TNL receptors through a mutation, the immune system of the plant was actually more active at all times. This heightened immune response in these modified plants meant that they were more resistant to pathogens and their associated diseases.
So we mutated these plants with a chemical called EMS. So it causes random mutations in the genome. And we isolate the plants with mutations that have the phenotype we want, which is enhanced disease resistance phenotype. So we're looking at enhanced disease resistance. We're looking for mutants that are more resistant to pathogens.
When a normal plant is exposed to a pathogen, it may result in disease in that plant and its eventual death. However, when plants with a TNL mutation are exposed to the same pathogen, they may be able to resist and fight off that pathogen. If these plants with a TNL mutation were used as crops, they could lead to a stable food supply without the use of toxic and harmful pesticides that have been proven to be toxic to both humans and the environment.
So the big picture is the plant immunity is super important for human beings because 30% of the crop is lost every year due to plant pathogens. So it's really important to understand how plants defend themselves.
These findings help plant researchers better understand how the plant immune system functions. Since the process of plant immunity is similar among most plant species, researchers hope to develop crops that are more disease resistant in the future. Applying the case of Arabidopsis in this study, scientists hope to solve the problem of food scarcity in different parts of the world by genetically modifying crops, thus improving the world's health and nutrition.